University of Missouri and a PhD in nutrition from The Ohio State University. Dr. Hackmann is an assistant professor here. His research attempts to improve feed efficiency of rumen microbial growth, and that's what he's going to be sharing with us today. So let's welcome Dr. Hackmann.
Thank you, Luis. Thank you, Francisco. Thank you, everyone, today. Thank you for the Milk Checkoff Committee for funding this work. Okay, so when we look at a cow, there are many things that we consider. We just learned that large body size may be one thing that we should not select for. But there's one thing that we oftentimes do not consider at all, and that's what lies within the microbes. So this is a video. We'll try it. Of some rumen fluid under the microscope. And what we see are protozoa swimming around. It's a short video, shorter than I remembered. So let's play it a few more times. Okay, swimming around. No? There we go. There are 10 million protozoa in a single ounce of rumen fluid. And they're harder to see, but there are things that look like dust particles. Those are bacteria. Let's play it once more. There are 10 billion, or 100 billion of those per ounce of rumen fluid. That's more than all of the people who have ever lived on Earth. Now, more than just a curiosity, these microbes really do have a great unseen importance. They're producing microbial protein, flows from the rumen, and when it's digested in the small intestine, it's accounting for over 50% of the protein that's metabolized by the animal. Now, microbes are providing a valuable service in producing this protein, but they could be doing better in the respect that they're inefficient. They're using only between 1/3 to 2/3 of the energy that they're generating for protein synthesis, for growth. I'm not going to show you a lot of tables of data, but I do want to highlight this one because it emphasizes how inefficiently they're growing. So this is the efficiency of rumen microbial growth for mixed microbes in vivo, mixed bacteria in vitro, and pure cultures in vitro, a number of systems. And when we look at their efficiency, we see these numbers here. For example, for mixed rumen microbes in vivo, we have 11 to 21 grams of microbial dry matter per mole of ATP. The significance is that we could compare this measured efficiency with a theoretical maximum. We can calculate how efficient microbes should be growing from biochemical pathways. And when we do that, we see that they fall far short of that theoretical maximum. It's only between 1/3 to 2/3 of that maximum, and that's the reason why we're saying that they use only between 1/3 to 2/3 of their ATP energy for growth. That means that there's a great potential for increased efficiency. If we approach the subject naively, we would say that we could increase it by 150 to 300% if we could somehow direct all energy towards growth. If we're more modest and we say that we want to increase efficiency by only 5%, we would still see great gains. We would increase microbial protein by about 0.2 pounds per cow per day. That decreases feed costs by more than 3 cents per cow per day. This is a back-of-the-envelope calculation where we're replacing RUP with urea. But no matter how you do the calculation, we see large improvements in efficiency. This translates to $1.5 million per year in Florida and $115 million per year in the US. So this gives us great impetus for trying to increase efficiency. Okay, so we mentioned that microbes are using only a small amount of their energy for protein synthesis. Where is the rest going? How can we try to improve efficiency? To do that, we have to look at the cell. We have a protozoan here, Isotreca protostoma. We'll see how it uses energy. So it takes feed. We have a terrible quality hay here, but it can be whatever the cow is eating. Ferments it and releases ATP. Some of that ATP is in fact used for growth. Again, 1/3 to 2/3 is used for growth, which is good. That's why we have the smiley face. But there are other sinks for that energy, and in the coming slides I'll show you what those sinks exactly are, but as a brief overview I'll just mention them here. We have maintenance, just like in the cow. We have reserve carbohydrate, where carbohydrate is being stored within the cell. And we have something called energy spilling, which is burning energy for the sake of burning energy. These microbes are just burning off excess energy as heat. Okay, so again we'll go through each of these sinks. Maintenance is simply the cost of living. For example, cells need to maintain ion gradients. They typically have a high concentration of sodium outside and potassium inside, and they have to maintain these ion gradients. A good indication that a cell is dead is if these ion gradients no longer exist. This is needed to keep the cell alive, but the net product of this process is heat. It's energy that's not going towards growth, so it is decreasing efficiency. Because maintenance costs are unavoidable, it's a fixed cost. I'm not going to be talking any more about it, really. I'm going to be focusing on the other energy sinks that I think we can do something more about. One is reserve carbohydrate. This can account for up to 50% of the cell mass. We have a protozoan before feeding, and we can see it's transparent. After feeding, it's packed itself with so much reserve carbohydrate that it's now opaque. Now, this may seem like an economical way to handle energy. We're storing it, but this storage is inefficient. It requires irreversible expenditure of ATP. Up to half of the ATP that will be released during fermentation of a glucose molecule can be irreversibly spent on the storage of that glucose molecule if it's in fact put in reserve carbohydrate. And last, we have energy spilling, which again, for the lack of a better definition, is burning energy for the sake of burning energy. So this is a phenomenon that's a bit outside of our everyday experience. So I have an experiment that will help explain the concept. This shows energy spilling in a rumen bacterium. A bacterium was dosed with glucose in a batch culture, and we can see the glucose was used rapidly over time, within about an hour. In response, the bacterium simply produced heat. It was burning off that glucose as heat. I don't show the data here, but it was growing very little. Okay, so we've identified what these sinks are. Besides the fact that they exist, what's the problem? These sinks have been seldom studied in mixed microbes from the rumen. The experiment, like I showed you on the last slide, is for a pure culture of a bacterium in vitro, and we can question how relevant are those studies to the rumen. Is energy spilling just a laboratory artifact, or does it really occur in the rumen? And as a corollary, the relative importance of these energy sinks in the rumen has not really been quantified. We would anticipate that reserve carbohydrate and energy spilling are competing sinks for energy. They're both responses to excess energy, but again, we don't know how important they really are in the rumen. Okay, so previous work that we've done has begun to address this problem. we showed that mixed rumen microbes, not just pure cultures of bacteria, but mixed rumen microbes that we take right from the rumen, are in fact wasting energy through both reserve carbohydrate and spilling. They're also directing energy towards maintenance, but they're wasting energy through reserve carbohydrate and spilling. So these are sinks that really seem to exist in the rumen. But there's an unanswered question. How do individual groups in the rumen respond? Which groups are responsible for reserve carbohydrate synthesis and energy spilling? So remember the video, we saw the protozoa swimming around. What is their response? And we saw the bacteria. You can see them a little bit more easily here. What is their response? Are they synthesizing reserve carbohydrate? Are they spilling energy? What is their potential? So the objective of our study was to quantify how protozoa and bacteria waste excess energy using an in vitro experiment, but with groups that were freshly isolating from the rumen to try to mimic the populations that we would see in the rumen, and with energy provided in the form of carbohydrate glucose. We're doing a similar experiment to here, but with microbes and not the pig. Okay, so our hypothesis was that compared to bacteria, protozoa would waste more energy via synthesizing reserve carbohydrate. And that comes just from a few slides ago where we saw those images of protozoa. Protozoan was packed full of reserve carbohydrate. Those kinds of studies are qualitative. We look under the microscope, we see that they seem to be full of reserve carbohydrate. What we're trying to do is confirm if that really happens using more quantitative methods. We also hypothesized that protozoa waste less energy via spilling. So our idea that reserve carbohydrate and spilling are competing energy sinks. If protozoa are wasting more energy via reserve carbohydrate, they are wasting less energy via spilling. Okay, so we took rumen fluid from one of two cows. We either filtered or we centrifuged the fluid. We filtered it, we got protozoa, we filtered out the small bacteria. As you saw in that video, the bacteria are a lot smaller than the protozoa. When we centrifuged the fluid, we got bacteria. We then dosed them with glucose, 5 millimolar. We measured their energy use, heat production by calorimetry. And we did chemical analysis for glucose, reserve carbohydrate, others, VFA, methane, hydrogen production. We measured a lot of different things. And we integrated all these measurements together. And in the end, we were able to say how much energy was being directed towards these sinks, reserve carbohydrate, spilling, and maintenance. Okay, so this shows how we separate the protozoa. As I mentioned, we're filtering them. You can see the cloth that we use for filtration there. Rumen microbes are sensitive to oxygen. They die almost immediately when exposed to oxygen, so we have to use this complex apparatus that excludes oxygen. So we work under completely oxygen-free conditions. Here is our calorimeter. We saw a photo of it before. It's about the size of 2 shoe boxes stacked on top of each other. It's like a whole animal calorimeter, just a lot smaller. It handles 1 to 2 milliliters worth of sample, so it's convenient for our experiments. We have a gas analyzer. We put the culture in here, and we can measure methane and hydrogen production in real time. So at the collection of this instrumentation, we can complete the experiment. Okay, so finally the results. This is an experiment when we dose the protozoa with glucose. And what we see is after we dose them with glucose, they're using that glucose rapidly, within an hour. They are synthesizing reserve carbohydrate in response. We can see that reserve carbohydrate is increasing and it's peaking at about the time that glucose is being exhausted. And we quantified how much was being synthesized. We found that over 50% of the carbon in glucose was being deposited in this reserve carbohydrate. Over 50%. So the majority of carbon is going into this reserve carbohydrate. So the protozoa really are synthesizing a lot of reserve carbohydrate, and that's an important energy sink. We also measured heat production, And we see that especially after dosing glucose, that heat production is increasing quickly. And this could be suggestive that they're also spilling energy, but we need to do a more thorough analysis, as I'll show you on the next few slides. When we did analogous experiments with bacteria, we see that they too are consuming glucose, though more slowly. Took them a couple of hours to exhaust the glucose in most cases. They synthesize some reserve carbohydrate, but far less than the protozoa. Only about 20 to 30% of the carbon in glucose is going towards the reserve carbohydrate. They're also producing heat, which again could be indicative of spilling, and we'll follow up with that in the next couple of slides. Okay, so how are we doing so far? We had hypothesized that protozoa waste more energy via synthesizing reserve carbohydrate, and we saw that when we measured reserve carbohydrate. We now have to look at whether they're wasting less energy via spilling. So I said they're producing heat. Maybe that's indicative that they're spilling energy, but now we need to look at those data in more depth. Okay, so this is heat production for the protozoa after dosing glucose. This is the same panel that we saw a couple slides ago, and we see that they're producing heat. In order to answer whether they're spilling energy, we need to start to determine what sinks are important here. So what we did first is we determined how much heat maintenance is accounting for. And we did this simply by extrapolating the heat production before dosing glucose. We extrapolated the baseline heat production. We then determined how much heat is released through carbohydrate synthesis. We did this through measuring the reserve carbohydrate, as I showed you, and we also measured fermentation products, and from biochemical pathways we could calculate how much heat should be released. And we can see that synthesizing reserve carbohydrate does account for a lot of the heat production. And then finally, the rest is energy spilling. And what we see here is that the protozoa are spilling very little energy. In fact, across experiments, there is no significant amount of energy spilling. I present this experiment that shows some energy spilling just so that you can see it here. But across experiments, it was not significant. Okay, with bacteria, we'll do the same. We calculated the amount of heat accounted for by maintenance. Reserve carbohydrate, we can see it's a lot less because they're synthesizing less reserve carbohydrate. And then spilling. We see that they're spilling a lot of energy. So yes, the protozoa are wasting more energy via synthesizing reserve carbohydrate, and they're wasting less energy via spilling. Okay. We'll skip over the next couple of slides because we're running a bit short on time, but we compared fermentation in the protozoa and bacteria as well. In the end, these data can potentially improve microbial efficiency and reduce feed protein. So that's a bit of a loaded statement. How do we go from laboratory experiments that we have here to feeding less protein? How do we bridge that gap? And the answer is through improved ration formulation software. Our experiments were specifically designed to improve ration formulation software. So let me explain. Ration formulation software predicts the amount of microbial protein. Here we see the Cornell model predicting bacterial protein, and it provides a good start for predicting microbial protein. But it still needs improvement. You can see how much scatter there is in this data. And the current experiments provide key data for improving this software. And it does it by giving a more mechanistic view of how microbes are using energy. So currently, ration formulation software represents these processes crudely, if at all. And again, these experiments fill the gap that exists in this software. Okay, to sum up, microbes waste up to 2/3 of their energy in the respect that they're directing only 1/3 to 2/3 of ATP towards growth. Both protozoa and bacteria are wasting energy, but differently. The protozoa are synthesizing reserve carbohydrate, whereas the bacteria are mostly spilling the energy. Measuring waste is a first step towards reduction. And our long-term goals, if I can speak, are to improve diet formulation software to try to increase microbial protein. Perhaps diet formulation software, once we plug in our values, will indicate feeding strategies by which we can improve microbial efficiency, and then to feed less feed protein. So I'd like to thank the people who are responsible for the work, the funding, particularly the Milk Checkoff, USDA, and CAPIs of Brazil. And I'd like to thank you for your time and be happy to field any questions that you have.
For the speaker.